Hello, everyone, and welcome to today's live broadcast, latest developments in high-titer transient protein expression using the ExpiCHO transient expression system, presented by Dr. John Zmuda, Director of Cell Biology, a Life Sciences Solutions Group, Thermo Fisher Scientific. I'm Julie Simmeroff, and I will be your moderator for this educational webcast presented by LabRoots and sponsored by Gibco, part of Thermo Fisher Scientific. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question in the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Please join me in welcoming Dr. John Zmuda. I will now turn the presentation over to him.
Thank you for the introduction, and good morning, good afternoon, and good evening to everybody on the line today. This is Jonathan Zmuda, Director of Cell Biology at our Frederick, Maryland site for Thermo Fisher, about 30 minutes due north of Washington, D.C. on the east coast of the United States. Unfortunately, I will have to apologize that my webcam, we had technical difficulties this morning, so unfortunately this will be a slides-only telephone conversation. What I would like to do is take you through some of the latest developments in our high-titer transient ExpiCHO system. And for those of you who have never worked with the system before, I'd like to go through each of the system's components and how they were developed and how they work in conjunction with one another to maximize the protein titers that you can obtain from each expression run. And then later in the presentation, we'll go through some applications notes around scale up and scale down of the system, as well as purification and other tips and tricks for optimizing the system and also ensuring that the system is running at peak performance for each one of your expression runs. So I'll start by going through the ExpiCHO system components first. And as with any protein expression system, the foundation of the system lies upon the cells themselves. And with ExpiCHO, we went back into our cell banks that we utilize for our GMP CHO-S cell line, and we went further back into the lineage to generate some diversity in those cells. And then we tested through clone picks the cells and picked high-expressing clones from those early CHO-S cell banks. So the ExpiCHO-S cell line is a completely natural subclone of the CHO-S cell line. So there's been no engineering to the cells in any manner. And these cells were picked based on a number of critical attributes that are necessary to have a good cell line for transient protein expression. So firstly, for transient protein expression, of course the cells have to be able to generate high titers of protein. And as we look at the specific productivity estimated for the ExpiCHO cells during what you would call log phase protein expression, we see that we have a picogram per cell per day productivity of about 30 ppd, which is quite good and rivals many stably expressing CHO cell lines. The second attribute that's critical for these cells is that they have a short doubling time, so that the cells grow rapidly. They're available for use when you're ready to perform your expression runs, and you're able to obtain a high-density cell growth, which allows for very high-density transfection in the ExpiCHO system. So the ExpiCHO cells have a very rapid doubling time of about 17 hours. So these cells will roughly triple each day when they're in log phase growth, and we'll show some more data along those lines, cell performance, in the next slide. And then thirdly, one of the other very key attributes to a good transient expression cell line is that these cells have a very non-clumping morphology. So if you were to compare, for example, the ExpiCHO-S cells versus typical CHO-S cells in culture media, in a growth curve type scenario, you would see that the CHO-S cells will get clumpy about a third to halfway through their maximum density cell growth. Whereas the ExpiCHO cells, and shown again in the bottom right on the screen, will continue to maintain a very nice single-cell morphology, even to quite high densities. So that is important because this allows for us to culture this particular clone up to high densities without the use of any anti-clumping reagents that would also be highly detrimental to transfection of the cells themselves. So these three key attributes really make the ExpiCHO cell line very useful for high-density, high-titer transient protein expression. So as I go through the next slides, I'll also talk a little bit about how to troubleshoot these cells and the cell growth, what they should look like out of thaw. And again, these cells are adapted for high-density culture. So in standard shake flasks, they will obtain approximately or greater than 20 million cells per mil during normal growth. These cells also have the ability, in conjunction with the ExpiCHO transfection reagent and other system components, to have a very high transfection efficiency, something that was previously attainable only in 293 cells that were expected to be an easily transfected cell line. Additionally, another key attribute for any cell line in transient expression system is stable growth and expression profiles. And what we'll show later in the presentation also is that the expression profiles for the ExpiCHO cells tend to be stable through about 15 to 25 passages, and that is also dependent upon, of course, how the cells are handled during that time, how they are maintained in culture so that the cells are not stressed from passage to passage. And then lastly, and we'll touch on this toward the end of the presentation, we wanted to ensure that the cells had, for example, CHO-like glycosylation profiles, so that the glycans coming out of the transient system match those very closely that were generated in CHO-S in a stable setting. So upon receipt of the cells, the ExpiCHO cells should recover very quickly post-thaw. And so in the upper right-hand figure, you'll see the thawing of 4 cell vials of ExpiCHO cells, and in blue is the viable cell density, where you'll note that from day 0 to day 1, in the first 24 hours, the cells will roughly double in density, and again double between day 1 and 2. And then by the time you're at day 2 to 3, these cells are already starting to take off into their normal log phase growth, and you can see between days 3 and day 4, the cells have really taken off and are growing very rapidly. In the red line above, you see the corresponding viability profiles. So you may see a very small dip in viability immediately post-thaw, but by day 2, the cells have already recovered to very high viability, 98+% routinely in subculture. And if for any reason the cells upon thaw do not appear to have these growth characteristics, that's something that it would be good then to bring up with our technical team, or also just to ensure that the cells had been, the chain of custody had been maintained and the cells had not thawed and refrozen, for example, at any stage upon receipt. So these cells should take off very quickly and be in a rapidly growing single-cell morphology immediately post-thaw. What we show on the right-hand side is ExpiCHO cells, and we're again looking at viable cell density on the left-hand y-axis, as well as viability on the right-hand axis. And these cells were seeded at 3 different densities, ranging from 0.125 to 0.5 times 10 to the 6 million cells per mil. And what you see is that in each one of these instances, we were able to obtain over 20 million cells per mil in shake flask cultures. So, of course, there are some differences in all counting measurements between instruments, hemocytometers, etc. So we say typically this is a good way that you can test for the ability of your cell growth conditions to be optimized by simply seeding the cells at one of these densities, typically 0.25 times 10 to the 6th, and allowing them to go out for 5 to 7 days and ensuring that you're getting somewhere in the ballpark of 20 million cells per mil. Again, give or take, assuming variances of 10 to 15% or perhaps even more between counting methodologies. So this is an excellent way to make sure that the cells are growing under the correct conditions in your laboratory. So the second part of the ExpiCHO system that is critical to allow us for these very high density and long expression runs to take advantage of the characteristics, the inherent characteristics of the CHO cells that make them a very good cell line for bioproduction, is the ExpiCHO feed that was formulated specifically to be matched with the ExpiCHO expression media, both of which are protein-free, animal origin-free, chemically defined formulations. And what we show on the bottom left-hand side is, for example, the ExpiCHO system has 3 different protocols that gives the user an option in terms of both the duration and the temperature of the protocol used. So ExpiCHO has 3 protocols, the first being the standard protocol, in which you have a single feed addition, and the entire protocol runs at 37C. And then we also have high-titer and max-titer protocols, in which you have a first feed addition, 18 to 22 hours post-transfection, followed by a temperature shift to 32C. And then in the case of the max-titer protocol shown on the left, we have a second addition of feed on or about day 5. And what we show here on the bottom left is the robustness in terms of the ability of the system to handle addition of the feed at different days. So if the feed were required to be added on day 4, that has no impact on the titer. Day 5 in the second bar from the left is where we normally recommend to add the second feed. And then day 6, you may see a very slight impact on titer, but even out to day 7, the cells will not crash, but it is that extra boost of nutrients that can really help those cells to push out to 12 to 14 days expression runs, where for very stable proteins you can obtain significantly higher titers. So the one other caveat, just for simplicity, is that the ExpiCHO feed does not need to be warmed prior to addition to the culture flasks. And in fact, in the next slide, I'll further give advice on simplification of the addition of the feed and enhancer simultaneously. On the bottom right, what we show is just one of the small changes that we implemented to the revised ExpiCHO protocol that was available online in December of 2016. So for those of you that have been using ExpiCHO for over a year, coming up on 2 years now, please do take a moment to download the new version of the protocol. It has a lot of additional information and tips for optimizing expression and generating the highest reproducibility possible. One small change that we made for the sake of simplicity was that for the standard and high titer protocols, each which have a single feed, we made them correspondingly both at a single feed of 24%. So now those 2 protocols are exactly the same as one another, with the exception of the temperature shift, so to alleviate any confusion there. And what you can see in the bottom right, again, is that even at lower percentages of feed, at 20% feed or 24% in the top 2 lines, we can maintain identical titers, no loss in titer, all the way out, in this case, to 12 days. Whereas at lower percentages of feed, shown in green, you will start to see the expression levels drop off a bit sooner without those added nutrients. And finally, in the red curve, at the 30% feed, we do see some impact of osmotic stress at 30%, which has slightly slower kinetics of antibody production. And so for all of these reasons, we have simplified the protocol so that both standard and high titer protocols now run with a single 24% feed. Very importantly to the ExpiCHO system is the ExpiFectamine-CHO enhancer cocktail. And this cocktail effectively greater than doubles the protein titers that we were able to obtain from the ExpiCHO cells. And once again, the enhancer does not need to be warmed before adding it to the cells. And importantly, in bullet 3, you see that the ExpiCHO feed and enhancer may be mixed together ahead of time, so that you're doing only a single addition to your flasks or multi-well plates. And in fact, this strategy was looked into for the multi-well plates in particular, so that you were not having to do multiple very small additions of enhancer and feed. And so on the bottom left, really a quite telling graph of why the utilization of all the system components together for ExpiCHO is critical to maximize the value and the performance of the system. If you look on the far left bar, if we simply transfect the ExpiCHO cells in their growth media, and we do not add enhancer and feed, the titers will be very low, so on the order of perhaps 1 to 200 mg per liter, which of course for CHO cell expression is still actually quite good. But in the middle, you see, so stepping back to that, what will happen to those cells is that, in essence, they will be transfected. They're very healthy after transfection. These cells like to grow, and they'll grow within the next day or so to their 20 million peak density and crash, and they will actually use most of their energy to grow instead of produce protein. So in the middle, you see when we simply add the feed to the system, so without the enhancer, in this case we can obtain much higher, about 5 times or 6 times higher titers than in the absence of the feed. And the feed has 2 different impacts on the cells. One is that it does have componentry that helps to slow the cell growth down, so that, again, the cells use their energy for making protein as opposed to growing. And secondly, it provides, of course, the nutrients for the cells to grow. And the feed has been developed with numerous proprietary components that allow for those components to remain stable at very high concentrations that are required for the CHO cells. But on the far right is where you see the true value of the entire system and the synergistic impacts of having a cell line adapted and matched to a media with a high-efficiency transfection reagent, a custom-tailored feed, and then an enhancer cocktail that allows these cells, again, to be very controlled in terms of their cell growth, but produce protein for many days. And you can see here that from using the feed alone to using the full system with the enhancer and the feed, you've more than doubled, almost tripled the titers that are obtainable from these cells. So all critical components of the system, and all in the manner in which the system has been developed as a system to provide the highest titers when all components are used in concert. On the bottom right, you see data from when we mixed the enhancer and the feed, and held it for either 1 day or 2 weeks at 2 to 8 C, or added it fresh in a sequential addition of enhancer and feed to the flasks. And so what you can see is that, and our recommendation typically is that on the day that you're adding your feed and enhancer, they can be prepared ahead of time and held at 2 to 8 C, and then again added as a single addition to your flasks or plates. And in fact, this formulation is even stable out to, we show here, 2 weeks at 2 to 8 C, no degradation in performance or titer from the system. So again, just a nice tip to save you from multiple pipettings. Go ahead and mix them ahead of time and add them singly to the flasks. Another very key component to the system is the ExpiCHO transfection reagent. And so what we show on the left in the bar graph is GFP, percent transfection efficiency, in XB293 cells, where we can reach approximately 90% transfection efficiency. Again, a very easy, known, easy to transfect cell line. Freestyle Max CHO, so the older CHO system, where we can get 40 to 50% transfection efficiency on the best day. And then ExpiCHO in each of the 3 protocols, where we're obtaining on or about 80% transfection efficiency of CHO cells at very high densities. So for XB293, the cells are transfected at 3 million. Freestyle Max CHO is at 1 million. And ExpiCHO is actually transfected at 6 million cells per mil. So this transfection reagent has very special characteristics that allow for high density, high efficiency transfection of CHO cells that has never really been possible previously for CHO cells. And there are a number of very key tips for using the ExpiCHO transfection reagent that I'll go through in the next couple slides to maximize both titers and reproducibility. And this is in part due to the special characteristics of this transfection reagent that allow for it to be a more powerful transfection reagent to transfect these high-density CHO cultures. So as a best practice, we recommend that prior to your complexation, that you dilute your plasmid DNA in cold OptiPro first, and that is very stable for long periods of time. And then when you're ready to make your complexes, you dilute your ExpiCHO into OptiPro, cold OptiPro, right at the time that you're ready to make your complexations. Immediately complex the plasmid DNA and the ExpiCHO, and I'll show in the next slide, but mix once or twice by pipetting or gentle inversion, and then those complexes are immediately ready to be added to your flasks. So ExpiCHO requires essentially no hold time or complexation time that I'll show in the next slide. But what I do show here is that allowing ExpiCHO to sit diluted in OptiPro for long periods of time can actually compromise the performance of the transfection reagent. The thought behind that being that because it is a stronger transfection reagent, that it may partly start to self-complex on its own with components of the expression media. So one of two ways. The second alternative path that works very well with ExpiCHO is that as an alternative to this, and what we suggest in the scale-down protocols for simplicity as well in the automation protocol, is that you simply doubly dilute your plasmid DNA with the entire volume of OptiPro that you would normally use to dilute the transfection reagent and the DNA. And then whenever you're ready to complex your DNA, you can simply add the ExpiCHO transfection reagent neat to your doubly diluted plasmid DNA. So in essence, a one-tube complexation reaction. And that has titers that are identical to the two-tube complexation reaction. So again, here, these are just some important tips for maximizing both performance and reproducibility of the system. And then what I show on this slide, again, is the best practices for the actual complexation reaction. So in the last slide, we talked about diluting the ExpiCHO with cold OptiPro when you're ready to make your complexes. And then here we're showing data that once you add your diluted transfection reagent and diluted plasmid DNA, in essence, again, you can mix a couple times by gentle pipetting up and down or inversion, and then immediately add them to your flask to transfect. So what we show here is less than 1 minute of complexation time. And up to 5 minutes of complexation time, you see no decrease in titers. And once you hold the complexes for more than 10 or 20 minutes, you will see some decreases in titer, likely again due to the complexes continuing to form and becoming slightly less able to transfect the cells. So while this is a bit of a paradigm shift in terms of transfection reagents, once you're used to doing so, we hear very positive feedback because in essence, on the morning of your transfection, you can grab all of your reagents out of the refrigerator, use them all cold, perform your complexation reaction, and literally within 5 to 10 minutes from the time you go to your refrigerator to transfect your cells are transfected. So it's a significant time savings in terms of transfecting the cells. And then when I get to the automation slides later, we'll discuss the ways in which we change the protocol a bit just to allow for the smoothest running on the automated systems. Again here, just bringing home the point that gentle mixing of the complexation reaction is the best way to form the complexes. So vortexing or very rough mixing by pipetting up and down 10 times very vigorously actually overforms the complexes, and while these are not toxic to the cells, you will not see a reduction in viability. You will see a reduction in titer. So again, upon complexation, 1 or 2 pipettings to mix or gentle inversion, and you're ready to transfect your cells. Another very interesting aspect of the ExpiCHO reagent is how little DNA we can use, even in light of the fact that we're transfecting 6 million cells per mil. So twice as high as XP293 and 6 times as high as most typical transfection protocols. So ExpiCHO typically is optimal in about a 0.6 to 0.8 microgram per mil range of plasmid DNA versus a microgram per mil, which is the industry standard for most systems. And in fact, even utilizing less DNA, you can still get very good titers. So the transfection reagent is very efficiently transfecting and delivering the DNA to the cells for the proteins to be made. What we show in this figure is actually a single concentration, a single volume of ExpiCHO transfection reagent, as noted in the protocol, complexed with different levels of plasmid DNA from 0.2 up to 1.2. And again, typically in our experiences, levels higher than 0.8 are not beneficial. And in fact, you can start to see slight viability dips at those higher DNA concentrations. When moving to the lower concentrations of DNA, 0.2, 0.4, or even lower, which we have evidence that suggests that for some hard-to-express proteins, significantly lowering the concentration of plasmid DNA can be beneficial, or in instances where you're limited in terms of your plasmid DNA or in large-scale preparations. And what we don't show in this slide, but when you do reduce your plasmid DNA to 0.4, 0.2, it is beneficial at this point, actually, if you're looking to go in that direction, to utilize a bit less of the transfection reagent, so reducing it proportionally to the 0.4 or 0.2, while keeping the entire complexation volume the same. And in doing so, those titers with the very low amounts of plasmid DNA will actually come closer to approaching the control titers that you see here for the 0.6 micrograms plasmid DNA and beyond. Lastly, with regard to the plasmid DNA, we and many others have found that the optimal heavy chain to light chain ratio for ExpiCHO is a 1-to-1 ratio, in contrast to the 293 cells that typically run at a 1-to-2 heavy to light chain ratios. The CHO cells do a very good job expressing the light chain, and thus a 1-to-1 ratio is consistently seen to be an optimal starting point for the ExpiCHO system. One of the new things that we're looking at that I'll just share today that's in the works is actually modulation of glycan patterns in ExpiCHO. And so we have developed some prototype reagents that allow for the modulation of these glycoforms in the ExpiCHO system. And why is this important? This is important because different CHO cells and different clones of the CHO cells maintained in different laboratories for many years in different media conditions and different feed conditions and expression conditions have differences in glycan profiles. And certainly for biosimilars, it is optimal to have your material, of course your transient material, be as similar as possible to your stable cell line produced material. And of course the first step there is using CHO cells for transient and CHO cells for stable. But some CHO cells and in different systems will have different glycosylation profiles. And what we show here is some prototype reagents that we've been working on that allows for us to modulate the glycan patterns of ExpiCHO. So in red we show the typical ExpiCHO glycan patterns where we're significantly G0F, which is very characteristic of CHO-S cells. And then through the modulation of different components of the culture media, we can reduce that level of G0F while bringing up the G1F glycoforms. And in fact, if you look at in terms of blue XB293 that has very complex carbohydrates, comparatively, we're actually able to change the ExpiCHO glycosylation profiles almost to look like a very divergent XB293 derived material. So while we would not necessarily be looking to make CHO glycosylation look like 293, it's very interesting that we do know that many users' CHO glycosylation patterns look more similar to what we show here in blue. And so the ability in the future to modulate these glycans should be of significant benefit certainly for biosimilar manufacturers. So I'd now like to go in the second half of the presentation and talk about key tips for optimal performance. With the system having been out for coming up on 2 years and many great collaborations and discussions with the ExpiCHO users, we have identified those key items that will ensure that the system is running at peak performance in your lab and have no questions in terms of whether or not you're getting all the protein possible out of the system. So as I mentioned previously, the ExpiCHO system has 3 different protocols, differing only in their feed amounts and number of feeds and temperatures. So if we look in green here, what we call the ExpiCHO standard protocol, in this case, the cells are transfected. 18 to 22 hours later, the feed and enhancer is added, and the cells are maintained at 37C throughout the duration of the expression run. For this particular human IgG1 antibody, on about 8 to 9 days in culture, we can obtain a gram per liter, which is comparable to what we can see in XB293 cells at about 6 days post-transfection. In the high-titer protocol, everything is the same as I mentioned as the standard protocol, with the exception that we shift the temperatures. And this hypothermic shift pays big dividends in the CHO system, in that on days 10 to 12, we can nearly double the titer, up to about 2 grams per liter. And finally, in the max titer protocol, where we break up the feeds so that we have 2 feeds, as well as 32C protein expression, we can then further bump up by about another third. So in the case of this antibody, very high titers, 2 plus, almost 3 grams per liter. And in the case of the max titer protocol, we can achieve that same gram per liter that we see in XB293 on day 6 using the CHO system. But obviously the big difference in the CHO system is that the CHO system allows us to go out and obtain 2/3 more protein over the next 7 days compared to the XP293 system. So XP293 is an excellent system. The 293 cells do a great job at making a lot of protein very quickly, but then they fizzle out, and they cannot maintain the productivity, the same productivity as the CHO cells can, whereas the CHO cells start off a bit slower and then ramp up and are able to make protein for very long periods of time, in this case all the way out to 14 days. So we do have recommended harvest times if we're talking about very stable proteins like antibodies. Typically in the standard protocol we harvest around day 7 to 9, probably day 10 to 12 for the high titer, and really 12 days is about as far as you need to go for the max titer protocol as the productivity drops off. And then of course, as with any protein in any system, considerations have to be given to protein quality, post-translational modifications, and ease of purification during the longer expression runs. So for example, in a very high titer, max titer expression run of an antibody, there will be more metabolic waste and buildup and cell debris in the cultures, even with very high viability toward the end of the cultures, which will make the processing of the supernatants a bit more challenging. And then, of course, looking at post-translational modifications for antibodies, it's always important to pick most correctly the protocol and the timing of harvest. Now, for non-antibody proteins or less stable proteins, the harvest days may look very different than this. So for membrane proteins, we may be looking at 48 to 72, maybe 96 hours. For other non-antibody secreted proteins, again, depending on their stability, it's really critical to do a time course of harvest and to understand where you're getting your best combination of titer and quality. So in this slide, this really summarizes in a nutshell what's going on in the ExpiCHO expression runs and how to troubleshoot the system. So in the top right, you'll see that the viable cell density, if we look at this, you can see at day 0, we've transfected the cells at 6 million cells per mil. A day later, we've added the enhancer and the feed, and these cells will typically, before we are able to control fully their growth, they will roughly double, maybe a little bit more in the first 2 days. But then you see by day 2 where the cell density is largely peaked. And then in the green line is the 37C standard protocol. So these cells will then maintain a density of somewhere between about 10 to 12 million cells per mil throughout the duration of the expression run. And in red, you see that this is added with the max titer protocol and the temperature shift. And so this really is a key attribute of how the ExpiCHO system works together. And I will say that to control the cells from overgrowing, it is absolutely a combination of the transfection reagent, the feed, as I had mentioned previously, and the enhancer. And again, this allows the cells to utilize their energy for making protein as opposed to growing. In the top right, you see a representative cell size graph for the ExpiCHO cells as they're expressing protein. The cells will go from 14 to 15 microns to as high as 20 microns during their peak expression levels, as you see in red here in the max titer protocol, when the cells are really pumping out a lot of protein. And then they'll stay relatively stable in that area. And then as the cultures wind down, you'll start to see the first notion of the cultures coming to an end is that the cell size will drop a little bit. The next day you'll see a drop in viability. So being able to monitor cell size is one of the first key indicators for ensuring performance of the system. In the bottom left is really the key attribute that tells you whether or not the system is running correctly in your lab and set up and optimized correctly. The ExpiCHO for a non-toxic protein like an IgG that we have here, and a very good control to use in the system is our rabbit IgG control plasmid that we offer, and that comes with the starter kits, that we know has very good similar viability profiles to what are shown here. So post-transfection, you may see a slight dip in the viability of the cells, a percentage or two. The cells recover then very quickly, and a viability curve that looks like this or similar indicates that the system is running very well. And I'll talk about troubleshooting in the next slide. But we typically would like to see viability 75% or greater at the time of harvest, And of course, depending on your harvest time, if it's earlier in the run, they may even be higher than that. That, of course, allows easier purification as well as less potential degradation of the product. And then on the right is just another example of the differences that you see in the titers between the two protocols. So in this slide, again, blowing up the viability figure, this is where we want to be at the end of the expression run, and I'll go in how to get there in the next couple slides. But in instances where the system is not set up correctly, and that may be, of course, everything comes down to your flask, the flask volume, the amount of volume that you're transfecting in that flask, your shaking diameter, and your shake speed. And of course, all of those things, need to be optimized. And in cases where one or multiple is significantly out of range, what we've seen is that on about day 4 or 5, you can see a reduction in the viability of the cells, indicating that the cells are not happy and some additional optimization should take place. So this is a pretty telltale sign. This is, of course, these cells are at very high density at that point. They're just coming off the stress of the transfection, the addition of the feed and enhancer, and when conditions are not correct, usually around day 4 or 5 is a telltale sign that the cultures are not going well. And when the cells start to die off like this, they'll make very little protein after that point. So it is critical to follow the recommendations in the protocol and to obtain this high viability during the expression run. So again, it may not always be this high. This is a non-toxic antibody. Certainly for non-antibody recombinant proteins can be different, but again, that's where you would tailor your harvest to your desired titer and protein quality. And again, using our rapid positive control antibody, you should be able to obtain viability plots similar to this, which indicates that the system overall is running at optimal levels in your lab. In terms of scalability, the ExpiCHO system is scalable from 125 to 2 liter using the same conditions, and I'll break these down a little bit further in the next slide. And then once moving to the 3 liter shake flask that has a very different shape to it, typically we're operating closer to the 75 to 80 RPM range for the 3 liter shake flask. And I will, in this slide, these are indicative of some of the data empirically derived that has been added to the ExpiCHO protocol and updated. So this really is the key to the entire system, and matching the flask size on the top with the volume to transfect in the first row. And this gives you these media to flask volume ratios that you see running across, so very scalable from 125 to 2 liters. And depending on the volume in the 3 liter flask, they will range from 1 to 3 to 1 to 3.5. So above 750 ml starting volume in the 3 liter flasks is not recommended without optimization. So you can go 5, 6, 7, 750, no problem. And then, of course, matching this to the shake speed. So this is critical. The most common shaking diameters are your 19, 25, and 50 millimeter shaking diameters. And for the 125 to 2 liter shake flask, we're recommending 125 for the 19 millimeter shakers, 120 for the 25 millimeters, and 95 for the 50 millimeter orbit. And on the right-hand column for the 3 liter flasks, again, these change quite dramatically, 75 RPM for the 19 millimeter, and actually 80 RPM both for the 25 and the 50 millimeter shaking diameter. So again, those were empirically determined as those numbers, the theoretical calculations would not have led to these results. So these have all been tested empirically. And then you see, in the case of the flask type, we're recommending vented, non-baffled across the board. I will mention a couple instances where the baffled flasks are superior in the next slide. And then what you see here, the final culture volume, once the addition of all feeds and enhancers, ranging from about 35 mLs up to 1 liter in the 3 liter flask size. So in one instance, or actually 2 instances, the baffled flasks we found have worked very well for the ExpiCHO system. At the 125 ml scale and the 250 ml scale, the baffled flasks actually work quite well and will further enhance protein titers. But there are a few changes that you need to put into place, very minor, if using the small-scale baffled flasks. Above the 250 ml scale, we do not recommend the Baffled flask. There is no benefit in using them. But what we do show here is that in the instance of 125 ml flask size, where in this experiment there was a bit of variability using a 19 millimeter orbit and the 125 ml flask size. So in black you see the non-baffled flasks, and red you see the baffled flasks. And even by day 5 you can start to see the baffled flask showing higher titer. By day 8, so in this case the cells had some stress to them, as I showed in the viability plot, and 3 of these vessels actually have largely stopped making protein, whereas 2 still look quite good. And in red, for the baffled flasks, all of them are performing very consistently and again, even higher than the best non-baffled flasks. We see the same thing all the way up to day 12, where the non-baffled flasks, again, these 3 flasks have not made much more protein, and very tight in the upper right-hand corner is the baffled flasks for the 125 ml condition. In general, we've only recommended this for the 19 millimeter shakers that have seen some variability, the baffled flasks. So there's 2 ways to handle that. One is to speed up the shakers to 140 RPM, and this again is captured in the troubleshooting notes of the protocol, or to move to the baffled flasks. The second potential benefit of the baffled flask at the 125 ml size is that it does allow you to increase your transfection volume, in fact, all the way up to 50 mLs. And on the right, you see that in doing so, you simply have to match the speed for expression to the volumes in the baffled flasks, as shown on the right here. And as you have more volume, the speed has to go higher. But in some instances, that can be very beneficial. The ExpiCHO system also works very well in the mini bioreactor or spin tubes. And what we show here is just compared to the control flask, a starting volume of 5 mLs to 20 mLs, standard protocol, and these are the final volumes once the addition of the feed and enhancer. So very comparable to what you see in the shake flask, and then the high titer protocol out to day 10, again, very comparable to what you see in the shake flask. Shown above, and these are captured also in an application note. In this instance, we were using 240 RPMs on a 19 millimeter shaker with about a 30-degree angle once you get to 10 mLs or greater. So at 5 mLs, these tubes can still be vertical. Once you get to 10 mLs or greater, in order to keep the cells in suspension, you do need to angle them, as shown in the tube rack here, at about 30 degrees. Similarly, the system works very well in the 24 deep well plates. So we recommend the rectangular bottom or the pyramid bottom plates. The round bottom plates also work comparably. We've just seen some better robustness in terms of broader ranges of shake speeds that the rectangular bottom plates are able to provide consistent results. So in the 24 deep well plates, these are very popular, about a 3.5 ml final volume. Again, these are captured in an application note. This final volume can be changed a little bit up or down, again, based on shake speeds. And we recommend in general, if you're not working with one of the good high-ends, for example, the Kuhn Duet system, the plastic plate lids seem to be superior in terms of evaporation compared to the gas-permeable plate sealer tapes. So we're simply using the blocks with the plate lids and putting them into humidified incubators and leaving them undisturbed during the duration of the expression run. Again, shown in red is the control flask versus the deep well plates for the standard and high titer protocol on the left figure. And again, on the right, this is the protocol for the 96 deep well plates. So here again, using the plastic plate lids, 850 to 950 RPM on a 3 millimeter orbital shaker. And the orbital part of this is very important. So some of the 3 millimeter shakers will be linear, for example, for ELISA plates or similar. It is important that it is a 3 millimeter orbital shaker. And here we can obtain about a mil of final volume in the 96 deep well plates. And then lastly, one of the things that we've been working on and have shared in certain instances, in comparison to the 293 cells, 293 cells are very inflexible in terms of what they will allow you to do. They like to have a single protocol. Again, they make protein fast. They die off fast. The CHO cells, once you have them set up well in your lab, they're much more flexible, and the system actually becomes a bit more of a mini stable cell line production system. And so in this case here, what we actually show is that for instances where you may want to get maximum protein as fast as possible, we do have recommendations that we can provide for ultra-high density transfection, so transfecting the cells at 10 or even 12 million cells per mil. And what's shown in the blue curve is that transfecting at 10 million cells per mil, you can see how much faster the titer comes up compared to transfecting at the standard 6 million cells per mil per the kit protocol. So this, can be very useful for quick expression runs where you're looking, in 4-day window or a short window to come up with some preliminary results. And again, we can provide certainly suggestions on how to get the cells, in a healthy state to allow for good transfection. We, in general, have to, in this case, increase the volume of the DNA expifectamine complexation reaction up to 3 mils. So we're needing to add about 1.5 times as much DNA for the higher cell density. And again, this is very beneficial for quick screens and potentially for toxic or membrane proteins if you're, looking to express them very quickly at small scales. So this can increase your volumetric productivity. Of course, at the same time, with this many cells, you're going to have more metabolic waste, and at later times in culture, the 10 million cell condition will start to taper off. The cells will stop making protein, and the 6 million cells will eventually catch up to them. So again, this is a strategy for much shorter duration expression runs. So we'll talk just a little bit about expression data, and I know this table is probably a bit hard to see, but I just wanted to highlight these are data from external labs in which we have documented fold increases comparing the ExpiCHO system to the current system that they were using, and you can see in many instances these are can be double digits, 4 or 5-fold more. Significantly, for example, in this case, greater than 3 grams per liter for this particular antibody on the left-hand column. But I think importantly also is the instances where in the middle column, 2 human antibodies could not be made in the XB293 system or precipitated, whereas ExpiCHO, shown in red, was able to give 1/3 to 1/2 gram per liter. And again, on the right-hand column, for some bispecific molecules, the CHO cells seem to do a very good job of expressing these sort of atypical type proteins. And again, in red, you see 2 bispecific antibodies that could not be made in the previous CHO system, and they were obtaining, again, over 1/2 gram to 3/4 gram per liter in the ExpiCHO system. And what we've seen in general, ExpiCHO seems to do an excellent job for expressing murine antibodies, well, antibodies of all species, but murine antibodies in particular compared to the human 293 cells. And then in this slide, we show a direct comparison of the ExpiCHO system to XB293 for a panel of 20 rabbit monoclonal antibodies. And what you see here in blue is the ExpiCHO titers, a mean 2.5-fold improvement over XB293, higher for each one of the antibodies expressed. And again, for MAB19, we had a couple hundred mg per liter of this antibody in ExpiCHO, nothing detectable in XB293, and then 1 antibody that, for whatever reason, did not express in either system. So again, I think at the end of the day, each of these expression systems has its own benefits, pros and cons, and working in concert between XB293 and ExpiCHO largely ensures that you will be able to make the protein, the particular protein that you're looking to express in one of those systems. Some work here just shown in terms of FAB generation in ExpiCHO. Does a very good job of generating FABs. Here we have C and N-terminal histag proteins, and then we are currently in a number of collaborations expressing membrane proteins in ExpiCHO, both GPCRs, ion channels, etc. And to date, we've seen very good expression of membrane proteins and very good activity of those proteins in ExpiCHO compared to either bacteria or 293-based systems. So another good alternative for membrane proteins as well. And again, in those instances, thus far, we've used lower amounts of DNA, around 0.2 to 0.4 micrograms per mil, to get very good expression of membrane proteins in about 2 to 4 days. Here I just wanted to highlight a publication recently that moved from 293 cells to ExpiCHO to make these very large HIV nanoparticles for immunization. And In the initial testing of ExpiCHO, they were able to generate a 20-fold increase in particle yield from ExpiCHO to XB293. And then we actually met with this lab and provided a couple of additional tips and moved to the temperature-shifted max titer protocol. And they were actually able to increase that to a 60-fold increase over 293 cells. And even more importantly than that is that the ExpiCHO material, derived material, was a very clean single peak by SEC, whereas the 293 was always multiple peaks, which required an upfront additional processing step in the 293 cells, whereas ExpiCHO actually allowed for this very nice single peak of this very complex nanoparticle to completely eliminate the upfront purification that was having to be done with the 293 cells. So a really interesting case study on a very hard to express protein complex. As a proof of concept, we automated ExpiCHO on the Ambru system, and in the interest of time, I won't go into this too much, but suffice it to say is that the only difference in running ExpiCHO on the automated system versus the 293s is the transfection complexation. And so in that case, again, we simply doubly dilute the plasmid DNA, and then we allow the robot to add the neat exfifectamine CHO reagent to the plasmid DNA, mix twice, and then inoculate the vessel immediately thereafter. So just a slight difference in programming, but ExpiCHO runs very well on the automated systems. Just a note on purification. So all of these expression systems are different. They have different components in them, and they behave somewhat differently in terms of both clarification, filtration, purification. Really not surprising. We generally recommend for ExpiCHO, because the supernatants can be a bit gooier, if you will, because of the long expression runs and the high cell densities. We recommend 5 to 8,000 times G spin step to really pellet down a lot of the components, the non-protein components. And this allows for much easier filtration through the 0.22 micron filters. Also, depth filters do a very good job at handling the supernatant. One key difference, and this is also in an application note that's online, is that the ExpiCHO supernatant does a better job being purified with either tris or HEPIS buffers using the MabSelect Shore columns, protein A columns, rather than the phosphate buffers. And we do call this out in the purification optimization note. And then again, for ExpiCHO, ion exchange polishing step, CEX, AEX also will do an excellent job, getting to 99% purity of the antibodies. But this one difference in buffer is quite important, as almost all protein A resins recommend using phosphate. And if phosphate is something that you need to use, there are other strategies that we can share with you for best purification in the phosphate buffers. But in general, tris and HEPIS have been shown to be very effective at good recovery and good cleanup of the protein. In terms of characterization, I'll just take a minute to show some gels here, non-reduced and reduced gels for ExpiCHO IgG in a standard high or max titer protocol. Expressing at 0.6, 1.1, and 2 grams per liter versus stable CHO-S and versus XB293. So very consistent post-protein A purification profiles. If we look at SEC for ExpiCHO, standard titer, harvested in the top panel at day 5 or day 8. In the middle panel, high titer at day 5, day 11. And in the bottom panel, day 5 and day 14 for the max titer protocol. You can see that in each case we have a good monomer peak just past protein A purification, and consistently about 4 to 5% high molecular weight aggregate, regardless of the titer or the expression protocol used. And then once we have further cleaned these up with ion exchange, we can easily see 99% plus purity of the antibodies. Lastly, I just wanted to note glycans. So if we look at the same molecule expressed in ExpiCHO, a stable CHO-S cell line, or XB293, we see the significant differences in the glycosylation profiles. So stable CHO-S and ExpiCHO in the top and middle figures show very similar profiles with primarily G0F glycoforms, whereas in the bottom panel, XB293, you see much lower G0F and more G1F glycoforms for XB293. So again, as I mentioned in the very first slides, we see glycosylation patterns between stable CHO-S or GMP cell line and ExpiCHO that are very similar. Again, however, these may be different than other CHO cell lines maintained in different medias, different feed, and different production conditions, which is why the potential to modulate the glycans is important going forward. As I mentioned, there's 3 application notes online. On the left is just the essential protocol guide with many FAQs, many of the things that I've talked about today. In the middle is the application note for scale down of the system, 24, 96 deep well plates, as well as the 50 ml spin tubes. And on the right is the guidance for protein A purification in the ExpiCHO system. And lastly, there have been a number of papers, but I call out 2 in general. One was very early work, very nice work done at XXII, comparing the ExpiCHO system to 293 cells and other expression systems. And on the right is the paper that I mentioned recently published about these trimeric HIV antigens, nanoparticles expressed in the ExpiCHO system, with another publication on this coming out shortly. So I just wanted to take a moment and thank the team that has worked on and continues to work on ExpiCHO to support different needs and applications. Xiaoyan Liu, the lead scientist on both the XB293 and the ExpiCHO systems. Jian Liu, who's responsible for the cell line engineering, the scale up and scale down molecular biology. Wendy and Kyle that have been working diligently on scale up, as well as the Ambru bioreactor. So what I did not have time to go into today is some of the wave scale-up work that has been completed up to the 10-liter scale. So we hope to have that in an application note in the coming months, as well as Melissa Cross, who is our on-site media engineering expert, and Lena Mpanda, who does our protein purification and characterization. And so with that, I will stop. I know we're close on time, but if there are any questions, we can answer them now. And of course, there are other mechanisms through ExpressionWorld to have those questions addressed as well. So thank you very much for your time.
Thank you, Dr. Zmuda, for that very informative presentation. It's time for Q&A. If you have a question you'd like to ask, please do so now. Just click on the Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the Send button. We'll answer as many questions as we have time for. If we are unable to get to your question, you will receive a follow-up email after the presentation. Let's get started. The first question is, what are the most important considerations as we get ready to use ExpiCHO for the first time?
So great question, and again, going back in our slide deck to matching. So firstly, ensuring that your cells are growing as expected. That's the most important thing for any of the protocols, that the cells are thawing well, growing as expected. And then secondly, again, it is matching the conditions from the shake flasks with the volumes to be transfected, and understanding your instrumentation, the shaking orbits, and the speeds. So I think as you proceed to get the system started in your lab, please do read through the protocol, follow those steps in there. They, again, have been empirically determined for optimal performance. Ensure that the cells are growing well and within the ranges provided in the protocol, and then ensure that you're following the recommendations in the table that we have here. And really that first step, the key of matching speed and volume to flask ratios and orbiting diameter is absolutely critical for getting the system up and running correctly.
Great, thank you for that answer. And we'll wrap up with one more question, and that question is, what are the most important considerations as we get ready to use ExpiCHO for the first time?
I think that's a repeat question.
I'm sorry. What is the best way to verify that the system is running optimally once we start using it?
Great, and so I'll back up again on my slides one more time. This is a key, this slide is very critical, and as I mentioned here, the best way to ensure that you're getting the protein yields that you should be is to utilize our positive control rabbit IgG antibody and ensure that your viability profiles are high all the way through the expression run and to the time of harvest. And that will indicate that all of the conditions that I just mentioned in the previous question are optimal, and that you should be getting optimum performance of the cells. And I do see there are a lot of questions in the queue, and I do promise to get to all of them following the ExpressionWorld, but because we're out of time right now, we will answer those offline.
I would like to once again thank Dr. Zmuda for his presentation. I would also like to thank LabRoots and Gibco, part of Thermo Fisher Scientific, for making today's educational webcast possible. Additional questions will be answered and published on Thermo Fisher Scientific's website. You will receive an email notification when answers are available. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through April 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you soon. Goodbye.